we call it polyribosome. So it looks like this. You have multiple. So the polyribosome looks like this. You have multiple ribosome on the same messenger RNA. So this is the same messenger RNA. You have 5 ribosome on the same messenger RNA, and then they do the translation at the same time. So I told you that there are 3 sites on the large subunit. You may not be able to see it. Let me use a different color. So it's the E site, P site, and A site. So during translation, codon on the messenger RNA, this is very important. Base pair with the anticodon, which is on the transfer RNA. Transfer RNA carries specific amino acid according to the anticodon code. The order of codon determines the order of transfer RNA. The order of codon determines the order of transfer RNA going into the ribosome. Because it determines the order of transfer RNA, the order of codon determines transfer RNA going into the ribosome. As you remember, transfer RNA carries amino acid. Therefore, the order of codon determines the order of amino acid. There are 3 steps in the translation: initiation, elongation, and termination. Only the first 2 steps require energy. Termination does not require energy. Well, then let's take a look at each stage really quickly. Initiation, okay. Bring the assembly together, all the component, meaning all the tools together. Initiation require all these. Small subunit capture the messenger RNA. Initiator tRNA, that means the start codon. So start codon usually is AUG. And AUG code for methionine. Methionine. And then of course we also have the large subunit that capture the transfer messenger RNA. So the initiation of the translation require at least these 4 components. And as I said, that the transfer RNA, the initiator transfer RNA has the start codon AUG, and the ribosome has the free site for AUG. P site, P stands for peptide, A site, A stands for amino acid, and E site, E stands for exit. So the first one should be the amino acid site. Somehow they arrange it into a weird order. Second one is the peptide site that links all the amino acid together. The last one is the escape site or exit site. The last one should be called exit site. So it allows the transfer RNA to go back to the cytoplasm and recycle. So the initiator tRNA, or we usually just call it start codon because it binds to the start codon. So the initiator tRNA, or the one that binds to the start codon AUG. I should use this one, the one that binds to the start codon AUG. Okay. Usually it first go to the P site first. So this is the initiation. Initiation, you can see that there's a small subunit capture the messenger RNA, and then you put the large subunit on top with the initiator tRNA in the P site. So it's like a hot dog, literally. The small subunit and the large subunit, they have the bun, and the messenger RNA is the hot dog. Okay. Elongation. Elongation, as the name suggested, it is when the ribosome move along the messenger RNA. It's like the engine moves along the railroad, then the smoke will come out. The smoke is the polypeptide chain. So we call this elongation. So elongation is a process by which polypeptide increase in length. Polypeptide increase in length is like the smoke coming out from the engine. But it occurs one amino acid at a time. It's not that smooth and fast. So we also have something called elongation factors that is required during the protein synthesis. Elongator factor helps the binding of the transfer tRNA, transfer RNA anticodon to the messenger RNA codon. So it helps the transfer tRNA codon bind to the messenger RNA codon. Why do we need a elongation factor? Why do we need to help the anticodon bind to messenger RNA codon? Let me ask you a question that we covered before. What are the attraction? Name the attraction between anticodon and codon. Let me give you 5 seconds to think about this. 5, 4, 3, 2, 1. Okay, time's up. The attraction that binds the anticodon to the codon we call hydrogen bond. Remember the DNA double helix? I told you that the bond in between the bases that hold the two strands parallel, those are the hydrogen bond. And then in here, why do we need elongation factor? Because we only have 3 bases on the anticodon and 3 bases in the codon. So they only have limited number of what we call the hydrogen bond. So if it is between A and U, okay. Remember, tRNA and mRNA, they both RNA. Whenever it is RNA, then we don't have T, we only have U. So A and U, they form 2 hydrogen bond. When we have 3 bases, that means 3 codon, 3 bases. That means you have minimum 6 hydrogen bond. Maximum, let's say they are all GC. GC, they form 3 hydrogen bond. So maximum, you have 9 hydrogen bond in a triplet coat. Okay, when the anticodon binds to the messenger, binds to the codon on the messenger RNA, you have 6 to 9 hydrogen bond in between them. Because of that, it is not very stable, and that's why they need something called elongation factor to help the anticodon bind to the codon. let's continue with elongation. Consists of 4 steps. So the tRNA will attach to the peptide. tRNA will with attached peptide is at the P site, and tRNA next amino arrive at the P site. Okay, this one is the initiator tRNA. This is the initiator. Only the initiator start at the P site. Only the initiator start at the P site. And the initiator has the, I already told you that it has the start codon, AUG, and the amino acid that carried by the this tRNA is coded for methionine. Methionine is usually the first amino acid in a polypeptide chain. And then subsequent amino acid or subsequent tRNA that carry amino acid, they all enters the ribosome at the A site. So only the initiator tRNA will go to the P site at the very beginning. But after that, all amino acid will go to A site. And then the last bullet point is very simple. It's just the tRNA when it is at A site, the polypeptide chain will transfer to this tRNA. So the polypeptide chain originally they are at the P site. Whenever we have the new tRNA goes into the A site, then the entire peptide chain from the P site will detach and goes to connect with the new amino acid at the A site. So this is like the sequence of elongation, okay, very straightforward here. So as you can see that originally you have the polypeptide chain in the P site. The peptide chain is at the P site, and then new tRNA with the anticodon that match the next triplet code. Then they will be collected by the large subunit at the A site, of course. And after this, the entire polypeptide chain will shift and move to the entire chain will attach to the amino acid on the A site. And after this, the entire hot dog or the entire messenger RNA will shift so that the original one that was from in the P site, the start codon, the initiator mRNA, they will leave as soon as you hit the E site, and then the entire process just recycle and continue.